Hello, welcome to the second practice midterm for CHEM271. This will be going over topics covered on the first midterm, so protein structure, acid base, protein purification, bioinformatics, and hemoglobin. So we will start with question 1. Question 1 is, why is proline the only amino acid that has an equal balance of cis and trans peptide bonds? So I already wrote this out. Essentially, so here is a model of what proline looks like. So rotation around this carbon nitrogen phi bond is restricted in proline, and that is because proline has a cyclic ring structure, as we can see here. So generally, in the peptide backbone, the trans configuration is stable, so it sort of is a zigzag within the backbone. However, in proline, because it's restricted due to the cyclic ring, the cis configuration can be produced and can be favorable. So this is sort of what a trans looks like, while this is sort of what the cis would look like. So the cis would have these clashes here, while the trans has less clashes. So normally the trans is favored for pretty much every other amino acid, but for proline, because of this cyclic R group here, the cis configuration can be favored because proline is fixed in this configuration. So it's fixed in either the cis or trans configuration. This is what the cis configuration looks like. You can sort of see clashes here between the peptide backbone, like here, and the inflexibility will stabilize the cis configuration. And in order for the configurations to be swapped, an enzyme is actually needed, prolyl isomerase, so therefore it is sort of stuck in either the cis or trans configuration, and because it's stuck in the cis configuration here, it is stable in the cis configuration. This can lead to turns in the backbone, which allows for turns in the peptide structure. Normally glycine and proline are the only amino acids that can do turns. Glycine because it's small, and proline because it can form cis configurations that are inflexible and more stable, versus normally the peptide backbone, which is more of a zigzag trans configuration. All right, the next question. How does hydrogen bonding network between amide and carbonyl groups differ between alpha helices and beta sheets? So let's first look at how hydrogen bonding works within the alpha helix. So here, the hydrogen bonding will occur between amino acids in the same chain. So between amino acids in the same chain, specifically between amino acids I and I plus 4. So for instance, this could be amino acid I, and this could be amino acid I plus 4 here. Okay, and then the hydrogen bonding occurs within the alpha helix itself. So essentially it occurs between a carbonyl here and the NH here, so the amide here. So this represents our hydrogen bond. If we want to look at it in another perspective, so this is amino acid I, this is amino acid I plus 4, you can see it more clearly here. And the main thing to take away from this is that these are within the same alpha helix. They are just 4 amino acids apart. Okay? The other things to note here is that all of the carbonyls are going to point in the same direction, and all of the amide, the NH, those are all going to point in the same direction as well. So all CO in same direction, as well as all NH in the same direction. And what this means is that all of the dipoles are actually going to be in the same direction. So a dipole is when you have an electronegative atom, like an oxygen or a nitrogen, next to a less electronegative atom. And what that results in is a dipole where the electronegative atom will pull electrons away from the less electronegative atom, making, in this case, the oxygen or the nitrogen more negatively charged and the other atom more positively charged. So we can draw a dipole like this. So we put the little plus end of the arrow. This is our D+, that's the positively charged atom, and the more electronegative atom like oxygen or nitrogen gets the arrowhead. This is D-. Okay, and then we can do the same for this. So the hydrogen will be more delta plus, and the nitrogen more delta minus. So then we can see that hydrogen bonding occurs because the hydrogen here is positive and the oxygen here is negative. That allows for an H bonding. So we can see it here better, delta minus on the oxygen, delta plus on the hydrogen, and that's because of these two dipoles. And you can see how all the dipoles are pointing in the same direction. So all in the same direction. So what this is actually going to do is give the carboxy terminus a bit of a negative charge and the N terminus a bit of a positive charge. All right. So, and this allows for hydrogen bonding throughout the whole alpha helix, which stabilizes the whole thing. Okay. Now looking at beta sheets, we've already looked at this in the last practice exam. I'm just going to show anti-parallel here. If you want to see a comparison between anti-parallel and parallel, look at the last practice exam. So here, the hydrogen bonds occur between the two beta strands. So this is between beta strands. All right. And these are not all in the same direction. So if we draw these again, we can see we still have a delta minus here and a delta plus here connected. But if we draw them over here, we can see they are in different directions. So dipoles in different directions. So the main thing to take away from this is that in the alpha helix, the hydrogen bonds are within the helix. They stabilize it from the inside. The other thing to note here is that the R groups are all going to be pointing out from the helix. So that reduces steric interactions because all of the R groups are facing out. Okay? And versus the beta sheet, where the hydrogen bonds are between the two strands, the beta strands, and the dipoles themselves can be in different directions. So the next question is about coiled coils like alpha keratin. It's asking what is the sequence pattern in coiled coils. So the sequence pattern is a repeating motif of 7 amino acids, A, B, C, D, E, F, G. Okay? And what's notable is that A and D are always going to be non-polar amino acids, so valine, leucine, isoleucine, methionine as examples. E and G will be charged amino acids, and they will have opposite charges. So glutamate, arginine, lysine, glutamate being negative, arginine and lysine are positive. Okay? So what we'll see here is if we start drawing the amino acids, we can look at the positions between these two circles here that represent two different helices. So what a coiled coil is it's two helices that are sort of wrapped around each other and bonded. So if I'm just going to draw what that looks like, it's like two helices like that together. Okay? 1, 2. All right. So now what I'm going to do is I'm going to draw the sequence of 7 amino acids around this. And I am going to move a bit down the circle every time because there is a bit of a shift. They're not just next to each other in a circle. It's this, and then the next part in the helix is here, and then here. And then when we get to the next one, it's actually here, D, E, F, and then G is up here. So why did I do it that way? Well, it's because if we draw a helix like this, so A will be here, B, C, D, E, F, so something like that. So what we're doing is actually looking down from top, and we are seeing sort of a non-linear order here because of the way they are positioned here. So they are spaced out such like this. However, when we look down, we see D is closer to A because of their positions. They're positioned in similar parts of the helix, but they are still 4 amino acids apart. Okay? So now we can draw it for the other one. The other one will be complementary, but in a slightly different order. So A, B, so sort of reversed in a way. D, E, F, G. All right, so let's get into what we said earlier. So A and D are nonpolar. So that means this area right here is all nonpolar. So this is what we call the hydrophobic core. And this is made up of A and D. So this is in the center of the coiled coil, so right in between the two coils. And this stabilizes through hydrophobic interactions. Hydrophobic interactions. So what are hydrophobic interactions? So basically these are interactions between nonpolar molecules that will ultimately exclude water. So essentially they'd rather bind to each other than they would to water. Kind of think about how oil and water don't mix. So because of that, just naturally these nonpolar things would rather bind to each other. There also might be some van der Waals interactions in there as well, which are the temporary dipoles formed within nonpolar molecules. All right, so this whole structure is stabilized from this nonpolar core. The other thing is E and G. So let's say E is, say, positively charged. Maybe it's a lysine, and G is negatively charged. Maybe it is a glutamate. All right, and we can see here that we have interaction. So we have a positive next to a negative on both of these. So what can happen here is an electrostatic interaction. So electrostatic interaction. So the E plus and G minus in this case can stabilize through electrostatic interaction. So a positive charge next to a negative charge. And the last thing to note here is there can also sometimes be disulfide bonds. So those are covalent bonds between cysteine residues, and those can further stabilize the structure. So basically, these coils are very stably linked together because of the nonpolar hydrophobic core and the electrostatic interactions between the E and G charged amino acids that have opposite charges and that are positioned next to each other in the helix, all right? And in 3D space, sort of relative to each other. And that will be it for this problem. All right, question number 4. State if each protein is an ortholog, paralog, or homolog for FTP1. So we are given 3 proteins to compare to, right? So we are given FTP1, that's the protein we are comparing to. And then within cats, so this is a protein from cats, within cats we have FTQ1 and FTR1. So FTQ1 is a protease, FTR1 is an isomerase. And then we have from the hedgehog, HTP1, which is also a protease. So let's define these terms first. So homologs are members of a protein family, so just related from the same protein family. Paralogs are homologs within the same species, and these can have same or different functions. So these are products of gene duplication, all right? So same species with the same or different function. Orthologs are homologs from different species, and these have to have similar functions. So essentially, these are all homologs. They're all from the same protein family. So now we have to decide whether they are paralogs or orthologs, okay? So these are all from the same species, cats. So these must be paralogs. So this is a paralog, FTQ1, and FTR1 is also a paralog, right? So same species, they're homologs, and FTQ1 has the same function. It is a protease, but FTR1 is an isomerase, so it has a different function. That doesn't matter. Still a paralog because it's from the same species, same protein family as well. All right. Now looking at HTP1, so this is from a different species. This is from the hedgehog. However, it is still related, still the same protein family, same function. This is an ortholog. All right, so same function, but different species, but still a homolog, same protein family. So that's it for this problem. All right, question 5 asks us to draw and identify the binding curves for the full R and T state hemoglobin. So this is a 4-part question. This is part A. All right, so we have the hemoglobin curve here given. We have on the x-axis the partial pressure of oxygen and torr, And on the y-axis, we have gamma, which is fractional saturation of oxygen. So hemoglobin, remember, has 4 subunits, and the more oxygen that binds, the more likely it is to bind more oxygen. This is due to the conversion between R state and T state. So we know that R state equals high O2 binding affinity, and the T state has low O2 binding affinity. All right, and then as oxygen binds, we know that the R state becomes favored, so T state shifts to R state, right? So slowly the T states will start to shift over to the R state, more so as more oxygen binds to hemoglobin, right? So binding at one site, so hemoglobin is 4 sites, right? So binding O2 at one site favors these other sites going from T state to R state. Okay. So now looking at this, so let's draw what these two curves look like for full R state and full T state. So full R state would be all 4 in the R state, full T state is all 4 in the T state. All right. So if we draw what this looks like, so the full R state will look something like this. So it has a much higher ability to bind oxygen. So it kind of looks like the myoglobin curve because it's able to pick up oxygen very quickly at low partial pressures of oxygen, very high binding affinity for oxygen. The T state is much lower, so T state will look something like this. Okay, so regardless of partial pressure of oxygen, it doesn't want to pick up oxygen much regardless. All right. Now looking at the next question, which state does 2,3-biphosphoglycerate binding favor and why? So we should know that 2,3-biphosphoglycerate, 2,3-BPG, favors the T state. All right, and because of this, this lowers O2 affinity, so lowers O2 binding affinity, so it's less likely to bind oxygen. And this facilitates the release of oxygen to the tissues. So why does this happen? Well, this is 2,3-BPG here. And what we can see is that it has 2 phosphate groups, each with a -2 charge, and a carboxylic acid group with a negative charge. So this overall has a charge of -5. So that's a very negative charge. And what we see is if we look at hemoglobin, we have alpha-1, alpha-2, beta-1, beta-2. These beta chains in the center of hemoglobin, these have positive amino acid residues on them. All right, so something like this. This is just a visualization of this. And then when we have our 2,3-BPG here in the middle, it can bind to all of this because it is so negatively charged. Right, so the 2,3-BPG has a negative charge, and it is able to bind to this sort of cavity in between these 2 beta subunits to all of these positive amino acids. So this is a positive amino acid, something like glutamate, for example. All right. So essentially, the catch is, though, that this cavity here, so this cavity only exists in the T state. So 2,3-BPG is stabilizing the T state because it is binding to these positive amino acids in this little gap in the hemoglobin protein. But this cavity here, this only exists in the T state. So thus this can only stabilize the T state. And it makes it so that this conformation is very stable. All right? So it electrostatically interacts with 3 positively charged side chains from each beta chain in the center of hemoglobin and binds in a cavity that only exists in the T state. Thus, it is stabilizing the T state. All right, so next up, parts C and D. All right, now on to the next part of question 5, part C. In the context of pregnancy, why is 2,3-BPG important? All right, so let's draw up our normal hemoglobin curve again. So this is hemoglobin, and this is actually maternal. So maternal hemoglobin, it's just normal hemoglobin that the mother has. Okay, so the difference between fetal hemoglobin and maternal hemoglobin is that fetal hemoglobin has 2 gamma subunits, and these are instead of the beta subunits. And these gamma subunits only have 2 positive amino acids, which is unlike the 3 positive amino acids that we saw in the beta subunit, right? So these are what are going to bind to 2,3-BPG. And what this means is that if you only have 2 positive charges versus 3, so these gamma subunits bind to 3-BPG less strongly. So there's only 2 positive charges, so it doesn't bind the negative molecule 2,3-BPG as strongly, okay? So if it's binding less strongly, that means the T state is less stable. So if the T state is less stable, what does this mean? It means that the fetal hemoglobin curve looks different than the maternal hemoglobin curve. So it's actually above the maternal. So this is the fetal, right? And that is because the T state is less stable. So because the T state is less stable, it's more similar to the R state, in which it's more likely to pick up oxygen. So what will happen is the oxygen will flow from the maternal hemoglobin to the fetal hemoglobin. That is because the fetal hemoglobin has a higher affinity for O2. So fetal hemoglobin, higher O2 affinity because it's more likely to be in the R state. So because of that, this is what facilitates transfer of oxygen from maternal hemoglobin to fetal hemoglobin, supplying the fetus with oxygen. All right, next question. So why would 2,3-BPG increase cell sickling in sickle cell anemia patients? So sickle cell anemia, this is a T state change. Specifically, this is a glutamate turning into a valine. So a positive charge turning into a nonpolar residue. Okay? So because of this, this is a nonpolar residue. So what this results in is actually an aggregation of hydrophobic patches within these hemoglobins. So these hemoglobins are going to aggregate. Okay? So essentially we have a valine here, and this can bind to a phenylalanine here and a leucine here, and this can form a hydrophobic interaction that results in the aggregation of hemoglobin subunits. So, however, this interaction only exists when hemoglobin is in the T state. So, can only aggregate in the T state. All right. So now we can see why 2,3-BPG would be bad for people with sickle cell anemia, because it stabilizes the T state and encourages more of these hydrophobic interactions, more aggregation, leading to more cell sickling. Cell sickling is due to the aggregation of hemoglobin in these long chains that results in sort of a sickle cell shape red blood cell, something like that. And that's due to these sort of long hemoglobin fibers that result from the aggregation. And that's it for this question. All right, the next question asks, why does hemoglobin carry carbon dioxide to the lungs? So this all has to do with the Bohr effect. So essentially, carbon dioxide stimulates the release of oxygen at the tissues. Therefore, hemoglobin will pick up carbon dioxide at the tissues and carry it to the lungs. But why does carbon dioxide stimulate release of O2 at the tissues? So it's because when carbon dioxide dissolves in water, it produces carbonic acid. So carbonic acid is a fairly strong acid, and it will release a proton generating bicarbonate. So it will release this H+, and what this does is this will lower the pH of the blood, okay? And the H+ will actually end up stabilizing the T state. So this is for two reasons. Number one is the protonation of histidine 146 within hemoglobin. So this gives it a positive charge, and this positive charge can bind to a negative charge in aspartate 94. So this will form an ionic bond. And what this does, this will stabilize the T state, allowing the release of O2. The other thing that can happen is when the terminal NH2, so this is the terminal amino group, when this interacts with carbon dioxide, what this does is it can form this group. This group is called a carbamate ion. And this also generates H+, which continues to further stabilize the T state. But this carbamate ion existing, this will form a salt bridge at the alpha-beta interface within hemoglobin, and this will stabilize the T state. Again, release of O2. All right, so it's for these two reasons that H+ will stabilize the T state and will result in the sort of release of oxygen. This is what's known as the Bohr effect. So we can look at these three different binding curves here. So this is sort of the normal hemoglobin binding curve for oxygen, right? So this is sort of what we observe in the lungs. There's no CO2, there's not really H+, elevated pH, more basic. So we see a higher oxygen affinity, right? But in the tissues here, what we see is there's CO2 present because tissues are releasing this from aerobic respiration, and thus we have a lower, more acidic pH. So what this results in is lower oxygen binding affinity in the tissues, higher oxygen binding affinity in the lungs. Therefore, the tissues, they release oxygen, and they also have a higher CO2 binding affinity, so they are able to pick up CO2. And then at the lungs, it switches from the T state back to the R state due to the presence of the oxygen, and it is able to pick up oxygen and release the CO2. All right, so essentially within the tissues, we see a stabilization of the T state, while in the lungs, we see a stabilization of the R state. The T state has lower oxygen binding affinity, but it can pick up CO2, and in the lungs with the R state, high oxygen binding affinity, it's able to release the CO2. All right, so that's it for this problem. That is the Bohr effect. All right, question number 7. So this part A is having us give the type of interaction between a protein and a resin for a type of protein purification column, and also asking for one means of eluting the protein. So first up, we have ion exchange chromatography using an anion exchanger. So essentially, this is an interaction between a negatively charged protein and a positively charged resin. So remember, an anion exchanger is used to isolate a negatively charged protein. So that's what anion exchanger. A cation exchanger would be used to isolate a positively charged protein. Now, if it's isolating a negatively charged protein, that means that the column itself must be positively charged in order to bind to the negatively charged protein. So the charge on the resin is the opposite of what the name suggests. The name is suggesting what charge the protein has to be in order to be isolated. So for a cation exchanger, that would be an exchange isolating a positive protein with a negatively charged resin. For anion exchange, isolating a negatively charged protein with a positively charged resin. So how do we get our protein to be negatively charged? So in order for this to happen, we need to know the pI, the isoelectric point of the protein. This is the pH at which the protein has a neutral charge. So essentially, in order to get the protein to be negatively charged, we need a pH higher than the pI. At a high pH, a more basic pH, deprotonation is favored. So what this will do is it will make acidic acid or acidic residues like carboxylic acid negatively charged, and basic residues like amino groups, those will be neutral when they are deprotonated. So this is, for instance, COO minus and NH2 neutral. If we have an acidic pH, this will be protonation. So here we will get COOH neutral protonated acid residues, as well as NH3 plus, so also protonated basic residues. So this will give an overall negative charge at a high pH and a positive charge at a low pH. All right. So essentially, if we want our protein to be negatively charged, we need pH to be greater than the pi of the protein. All right. So then if we want to get rid of the protein from the column, we can use salt, or, I know it says give one, but we're going to give two. We can use salt, or we can use a buffer with pH less than pi. So why would we want a buffer with a pH below the pi? Well, what this will do is this will actually protonate the protein, so it'll give it a positive charge. If the protein has a positive charge and the column has a positive charge, that means that the protein cannot bind to the column. Thus, since it's not binding anymore, it will elute out of the column. So we can actually change the charge of the protein by changing the pH of the buffer we use. Or we can just use salt, which decreases the solubility of the protein, allowing it to crash out of solution, precipitate, and then it will leave the column. Okay, affinity chromatography. So this is using a nickel resin. Forgot to write that earlier. So using a nickel resin, so essentially affinity chromatography uses some sort of thing like an antibody or an ion to catch a very specific protein. So this will be an interaction between a his tag. So what a his tag is when you are cloning a protein, you can change the sequence of the gene to add a bunch of histidine residues to the end of a protein. And what that does is it gives sort of this long change of charge to the end of the protein. So this his tag will bind to the nickel ion in the residue. Okay? And these nickel ions are immobilized, so the his tag will bind to the nickel ion. Now, if we want to release the protein from the nickel resin, from the affinity chromatography column, we can add imidazole. So imidazole, what that will do, this will bind to the metal ions, to the zinc ions, and this will displace protein. So binds Ni, and this will displace protein. So this will allow the protein to release from the column, because normally it's the his tag on the protein that's binding to the nickel ion, usually through some sort of metallic coordination bond. And then if we add the imidazole, sorry, that will bind the nickel more strongly, thus releasing the proteins that are bound to the nickel. All right, on to the next question. So this question is asking us to just explain these results. So essentially, we have a technique. We're using gel filtration chromatography and SDS page, and we have the apparent molecular weight of, this is a hemoglobin. Right? So with gel filtration, we observe a molecular weight of 80,000 Daltons, and with SDS page, 20,000 Daltons. So why do we see different molecular weights for the same protein? Well, it's important to note hemoglobin equals 4 subunits. And these are 20,000 Daltons each. So essentially, 4 subunits, 20,000 Daltons each. This is 80,000 Daltons total for the quaternary structure with the 4 subunits, 80,000 Daltons, okay? So why are we seeing this difference? Well, the difference here is that SDS page, so these were both described in the first practice midterm, so I won't go over them again in detail, but SDS page has SDS in it. SDS will denature the protein, and it will also break interactions between subunits. So it will break apart subunits. So because of that, what we are seeing here is 4 subunits separately. right? And since we are seeing the subunits broken apart separately, we are seeing the sort of 20,000 Dalton subunits, not the entire quaternary structure. But with gel filtration, this has no SDS, thus quaternary structure is intact. So without SDS, the quaternary structure stays intact. All 4 subunits are still attached. Thus, we observe one larger protein with all 4 subunits together with a molecular weight of 80,000 Daltons. So the difference here is the SDS sort of denatures the protein and separates the subunits. Thus, with the SDS, we see the smaller subunits individually with 20,000 Daltons. But with gel filtration, there's nothing to split apart the subunits. So we see all 4 of them together with a larger molecular weight of 80,000 Daltons. Right? All right, this is the last question of the practice exam. This is an acid-based question, and it is quite long. So the first part of the problem says we have a solution, pH 7.4, and we're using a phosphate buffer with pKa equals 7.2. So we want to find the ratio of the conjugate base divided by the acid, the concentration of the conjugate base divided by the concentration of the acid. This is fairly straightforward. We will use the Henderson-Hasselbalch equation, pH equals pKa plus log of this exact ratio. Simple. So we have pH, we have pKa. So what we can do is find, as we showed in the acid-base problem set, we can subtract pKa from both sides, then take 10 to the power of both sides to get rid of the log base 10. So we can get 10 to the pH minus pKa is equal to A minus over HA. So in this case, we get 10 to the pH, so 7.4, minus 7.2 is equal to 1.58, and this is equal to the ratio. Simple enough. So does this make sense? So our pH is more basic than the pKa. So if the pH is higher than the pKa, we expect to see more conjugate base than we do of the acid, and that's what we see. We have a ratio above 1, meaning we have more conjugate base, thus the pH is a bit higher than the pKa. All right. Part 2, or part B, the concentration of lactic acid increases by 3 millimolars. Find the pH, and we are assuming that lactic acid is a strong acid, and that and we know that the total phosphate concentration will be 100 millimolar. All right. So the first thing we have to do is solve a systems of equations here, all right, before we can do anything else. And so we know already that the ratio here is 1.58. So we've already solved this. We already have one equation we can use to solve a systems of equations here. So 1.58 times HA, the concentration of HA, equals the concentration of A minus. We know that. We also know that the that the sum of the concentration of HA plus A minus is going to equal 100 millimolar. All right, so now what we can do is simply put this in here, and we get 2.58 HA equals 100 millimolar, and we can find that the concentration of HA is equal to 38.76 millimolar, and then we can plug that back into here, and then we can, if we do that, we can find the concentration of A minus. A minus equals 38.76 millimolar times 1.58, which is equal to 61.24 millimolar. All right, so now that we have both of those, we can now get started on the problem. So we have to go through the 3-step process as we did before. So the first step is get everything in moles, or in this case, we can do it in millimoles. However, the volume isn't given here, so we just assume what the volume is. In this case, I am just going to assume volume is equal to 1 liter. It's arbitrary, but 1 liter simplifies all of our calculations. So now so the first step here will be to get everything in millimole. So to do this, we just multiply everything by the 1 liter, and what we find is HA, we get 38.76 millimole. For A minus, we get 61.24 millimole. And then for our lactic acid, we can get 3 millimole. All right, now we have everything in millimoles. So now what we can do is enter this all into our Rice table. And because we have a strong acid, right, we are going to do stoichiometry. We're not doing equilibrium stuff yet. So this is all stoic. And essentially, because the concentration of lactic acid is the smallest of the three, we know that will be our limiting reactant. So this will fully react, right? So this is essentially H+. So in this case, we will have our A- here interacting with the H+. This is from lactic acid. And this will form the acid. So this reaction will go to completion because this is a strong acid. So essentially all of this 3 millimoles of H+ from the lactic acid will completely react with the A-. So now we start with I, which is initial. So we get 61.24 millimole, 3 millimole, and 38.76 millimole. All right, now we know that there's going to be a change, so these two are going to react, so the concentrations of both of these will go down. They will go down by the amount of the smaller of the two amounts. So in this case, they will both go down by 3 millimole. And this will gain 3 millimole, right? And that's due to the coefficients in the balanced equation. One of these plus one of these generates one of these. So we lose one part of this, we gain one part of this. So now we just do a simple bit of math. So we get 58.24 millimole, 0 millimole, because all of the H+ gets used up. And we add 3 to this to get 41.76 millimole. All right, part 3. We enter this all back into the Henderson-Hasselbalch equation. So pH equals pKa plus the log base 10 of millimole A- over millimole HA. As we found, you don't actually need the volume for this equation. It's the same volume for both, so therefore it will just cancel. So this makes this very easy for us. So pH is going to be equal to our pKa, so this is 7.2, plus the log of A-, so 58.24 millimole, divided by 41.76 millimole. And what we get from this is that our pH is 7.37. So because of this, we can see that we have a buffer here, and we have enough conjugate base present in the solution from the buffer that when we add this little bit of strong acid, the pH barely changes. But what happens if there was no buffer? What happens if we just add this lactic acid to solution? So because of this, to solve this, we just use pH equals negative log concentration of H+. Very simple. So we need the the H concentration in molarity. So negative log, to get to molarity, we do 3 millimole per liter, and then in a mole, there is 1,000 millimole. So we are essentially just dividing 3 by 1,000. And when you do this, you get pH is equal to 2.52. So we can see that without the buffer, the pH drops by a lot. It becomes very, very acidic because there's nothing to stop the H+ from entering solution, and it drops the pH very, very quickly. Versus with the buffer, the pH hardly drops at all because there's conjugate base present to react with the H+ and prevent the large change in pH.
And that will be it for the second practice exam, and it for the first part of Chem 271, all up to the end of the first midterm. Thank you for watching.